Good afternoon, students. Today in this lecture talk of question and answers, let us understand about molecular biology concept, what are nucleosides and what are nucleotides. Okay, these nucleotides and sides, which are the part of nucleic acids, right? So nucleotide are the main component of nucleic acid, which is also known as nitrogenous bases, right? So here, first one, nucleosides. Nucleoside is formed when one of the purine or pyrimidine base is attached to either D-ribose or beta deoxyribose is known as nucleoside. Okay, nucleosides means it's the combination of sugar and nitrogenous bases. It means sugars like maybe a ribose or maybe deoxyribose along with nitrogenous bases like maybe purine or pyrimidine. Okay, this is known as nucleoside. So what are the components of nucleic acids? The nucleic acid components are pentose sugar, nitrogenous base, and phosphoric acid. Here, phosphoric acid is missing. So in nucleosides, phosphoric acid has not come. Okay, phosphoric acid will not come. So only the combination of sugar and the nitrogenous bases are known as nucleoside. I hope I made this nucleoside clear. Next one, let us understand nucleotide. Nucleotides are the phosphoric acid esters of nucleosides. ಇಲ್ಲಿ ಯಾವ್ದು ನ್ಯೂಕ್ಲಿಯೋಸೈಡ್ಸ್ ಇರುತ್ತಲ್ಲ ಈ ಶುಗರ್ ನೈಟ್ರೋಜನಸ್ ಬೇಸಸ್ ಜೊತೆಯಲ್ಲಿ ಇಲ್ಲಿ ಏನಾಗುತ್ತೆ ಎಸ್ಟರಿಫಿಕೇಶನ್ ಆಗುತ್ತೆ ಎಲ್ಲಿ ಎಸ್ಟರಿಫಿಕೇಶನ್ ಆಗುತ್ತೆ sugar, that is along with the ribose or deoxyribose, there will be an esterification along with the phosphoric acid molecule. So what are nucleotides? Nucleotides are the phosphoric acid esters of nucleoside. So it's very simple to keep in your mind to write the answer in the examination. Okay. Nucleotides, what happens here? H3PO4, that is phosphoric acid, is esterifies with the one of the hydroxyl group of the pentose sugar. One of the hydroxyl group of the pentose sugar, that may be a deoxyribose or maybe a ribose. It means, whereas here, the hydroxyl group available for phosphorylation in RNA molecule are 2' carbon atom, 3' and the 5th carbon atom position. Okay, it means in the particular ribose sugar at the 2nd carbon atom and the 3rd carbon atom, 5th carbon atom position, there's the presence of hydroxyl group for the phosphorylation. Whereas in the DNA molecule, only at the deoxyribose molecule, 3' of the carbon atom and the 5th prime of the carbon atom, hydroxyl groups are there. But however, only the 5th carbon atom position is the most commonly phosphorylated condition. Okay, so at the 5th carbon atom of the ribose and the deoxyribose, there'll be a formation of the esterification, okay, with the phosphoric acid group. So this is about the nucleotide. Okay, I hope I made myself clear to you about nucleosides and nucleotide. Let us understand in detail here. So nucleoside means there'll be the presence of only nitrogenous base, maybe a purine or maybe a pyrimidine, sugar, okay, maybe a ribose sugar or maybe a deoxyribose sugar. The combination of nitrogenous base plus sugar is known as nucleoside. In nucleoside, phosphoric acid has not yet come. Okay, so position of the nitrogenous base to attach the ribose or deoxyribose is the first carbon atom. Okay, first carbon atom. So with this bond is known as glycosidic bond. And this is the 5th carbon atom. Okay, at the 5th carbon atom, there is an attachment of the phosphate molecule. That is esterification of the H3PO4. Phosphoric acid molecule esterification will happen. When phosphate molecule is attached here, then we can say this as nucleotide. When you say nucleotide, there are 3 components are going to come. One is nitrogenous base, deoxyribose sugar, then phosphoric acid molecule, that is a phosphate molecule. So all 3 components, when they come in touch together, then we can say nucleotide. I hope I'm making myself clear to you. Shall I go to the next slide? Okay, all right. The next one, in detail information about the nucleotide. Okay, the first one here, this is the base. Nitrogenous base are going to link at the first carbon atom. Okay, so at the 2nd carbon atom, That is, maybe for the ribose, hydroxyl group will be there. And for the DNA molecule, there will be a deoxyribose, oxygen molecules removed. So when we say nucleoside, when there is a base and the sugar, or maybe a deoxyribose or deoxyribose, or ribose means we say nucleoside. Okay. Once this particular phosphate molecule is attached, we can say that as nucleotide. So if there's a one attachment of the phosphate molecule, that is nucleoside monophosphate. Okay. Then second phosphate molecule attachment, nucleoside diphosphate. Third attachment, nucleoside triphosphate. Okay, together we can say this as particular triphosphates. Okay, always all the nitrogen bases we are getting in the form of triphosphate, ATP, GTP, right? TTP, UTP, like that. There is the presence of the three phosphate molecules. All right. Then the concluding part of the nucleoside and nucleotide. In nucleoside, there's the presence of pentose sugar and nitrogenous bases like adenine, guanine, cytosine, thymine, and uracil will be there. Okay, this is nucleoside, where you can see the presence of all the nitrogenous bases, sugar, or maybe a deoxyribose sugar. Okay, so this is nucleoside, adenosine, guanosine, cytidine, uridine, and thymidine. Sugar moiety may be deoxyribose or ribose sugar. So this is the nucleoside, and this is the nucleotide. The difference between nucleoside and nucleotide, only the here S, and this is the T. Okay, and in this nucleotide, this is the combination or components of phosphoric acid molecule, pentose sugar, nitrogenous bases. So the first one, nitrogenous bases here, then here the phosphate molecule will be there. Okay, whereas here adenosine, that is adenosine means what? This one, nucleoside. We do not say adenine. Adenine means only one nitrogenous base. Adenosine means this is the combination of adenine plus sugar. The sugar may be deoxyribose or ribose sugar. Together we say adenosine. So adenosine, when it come in contact, this is adenosine. What is that adenosine? Nitrogenous base plus the pentose. Together we say adenosine. So this adenosine, when come in contact with the phosphate group to the esterification, we say that as nucleotide. So this adenosine plus phosphate molecule, adenosine monophosphate. With the presence of other three phosphate molecule, we say adenosine triphosphate. Okay. I hope I made myself clear to you about nucleoside and nucleotide. Okay, this is the trick and the concept where you can keep in your mind about the nucleoside and nucleotide. Okay, so let us understand more detail about what is this nucleoside. Nucleoside is a nucleobase attached to a sugar molecule, usually a pentose sugar, a ribose, a deoxyribose. That is nucleoside, where you could see only the sugar and the nitrogenous molecule. Okay. Then this linkage refers to as a beta glycosidic bond. The linkage between the pentose sugar and the nitrogenous base. The linkage between the pentose sugar and the nitrogenous base is known as the glycosidic bond. This is the glycosidic bond. Okay. Beta glycosidic bond. The significant feature of the nucleoside is that if a nucleoside links with the phosphate group, eventually it becomes a nucleotide or a nucleoside monophosphate, which is the basic unit of nucleic acids. Okay, this is about nucleoside. Then coming to the nucleotide. So what is this particular nucleotide? Nucleotide is the building block of two crucial macromolecules, that is nucleic acids in living organisms called DNA and RNA. And they are the genetic material of an organism and are responsible for passing genetic characteristics from one generation to the next generation. Furthermore, they are important in controlling and maintaining cellular functions. Other than these two macromolecules, there are several other important nucleotides. For example, ATP, that is adenosine triphosphate, GTP are two important energy molecules. Then NADP, nicotinamide adenine dinucleotide phosphate, and FAD, flavin adenine dinucleotide, are nucleotides which act as cofactors. Okay. Also, nucleotides like CAM, that is cyclic adenosine monophosphate, are essential for all signaling pathways. Okay, so this is all about nucleotide. Then coming to what are the similarities between nucleotide and nucleoside. Both nucleotide and nucleoside have a pentose sugar and the nitrogenous base. When a phosphate group links with the nucleoside, eventually it becomes nucleotide. All right. So this is all about nucleotide. I hope I made myself clear to you all about the nucleotide and nucleoside. So first we went through about what are nucleosides, then about nucleotide, and then the representation, structure presentation. Nucleosides means nitrogen base plus sugar is nucleoside. When this along to this with this nitrogen base and sugar is the esterification of phosphoric acid molecule, then that will be a nucleotide. Okay. So this is all about nucleotide and nucleosides in detail. I hope I made this video clear. Thank you everyone for watching this video. Stay tuned with me for further more this type of videos. Thank you.